Hi everyone, welcome to the third part of the microorganism friend and foe. So we will learn about microorganisms. So just tell me what do you understand by fermentation. The previous class we have discussed this. If you can't tell, this will be your homework. Okay. Okay, I have shown the answer. Okay, let me tell. No need of homework. This is not your homework. Okay. So fermentation, what is the process? They make sugar. Any sugar is converting into what? Alcohol. That process is known as fermentation. Who are making this? This is caused by microorganisms. So write your answer like this. You will get full marks. Obviously. Okay. Now we'll see our friendly microorganisms that we will complete today. Food preservation and nitrogen cycle. Maybe nitrogen cycle I will create season 4. That depends on you. Okay. Now friendly microorganisms. A doctor prescribed. In the previous class we have discussed. What we have discussed, guys? Tell me to. First, that tell me. The previous class we have discussed about the what is the friendly microorganism helping in our food industry. Today we will discuss about the medical first medical industry. Let's see the doctor prescribed the tablets. These tablets are you all know and you all have taken, that is antibiotics. So we have already completed the commercial purpose, right? Now it's time for medicinal purpose. So let's go. So what are antibiotics? So antibiotics are the medicines that kill or stop the growth of disease causing microorganisms. Like any microorganism is causing a disease, this antibiotic will stop the growth of the microorganisms. And what is the source of antibiotic? That is also microorganisms. Means microorganism is only stopping the microorganisms. So this was that is they are stored in a tablets form, and that antibiotics that means that microorganisms are stopping the growth, preventing the further diseases. Okay. Now penicillin is the first antibiotic. It was discovered by Alexander Fleming in 1929. So Alexander Fleming was working on an the culture of disease causing bacteria. There he saw a mold. Okay. And he seen that um, the presence of this mold prevented the growth of bacteria. Means some bacteria was even killed also with this penicillin. The microorganism name is Penicillium notatum. That microorganism is being used. Then he come to idea of antibiotic that when these are entered in our body, means antibiotics are injected or taken, it slow down the growth of bacteria and prevents further disease. If someone is having AIDS, AIDS, no diseases, no cure is there. But what can an antibiotic do? It can stop it, but it will spread because this disease is very dangerous. Okay. I will discuss this about this AIDS in another class maybe. Okay. Most of the antibiotics destroy the cell wall of the bacteria and prevents their further cell division. आप जानते हो ये सेल है, ये न्यूक्लियस है, ये साइटोप्लाज्म है, ये सेल वाल होता है. अगर ये नहीं है तो what is present? Only nucleus and cytoplasm. When this nucleus and cytoplasm is broken, all the thing will flow out, and as the cytoplasm will flow out, the nucleus is slowly die. Okay. Antibiotics are used in humans, plants, everywhere it is used because it prevents the microbial infection. That's why it is used everywhere. Now, have you seen the polio is given to you? What is that? Is a vaccine or antibiotic? Vaccine. Okay, that is vaccine. So vaccine is the weakened microorganisms. इसमें weakened और killed microorganism होता है जो हमें disease होने से बचाता है, prevent the diseases. What happens? Let's see. सो वेन नॉट वैक्सीनेटेड, नॉर्मल कंडीशन आप सोचिए, कोई बाहर से कोई पैथोजन आया, मीन डिजीज कॉजिंग माइक्रो ऑर्गेनिज्म, वो हमारे बॉडी में गया, तब बॉडी में जब आया तब बॉडी ने एंटीबॉडीज रिलीज किया. एंटीबॉडीज लाइक वाइट ब्लड सेल, आपने पढ़ा है क्लास 7 में. एंटीबॉडीज ऑफ दैट विच फाइट द इन्वेडर, फाइट द फॉरेन पार्टिकल्स. This is a slow process. Body takes time to generate required antibodies to fight against. Body को पता नहीं है कि ये कैसे हम लोग इस एंटीबॉडीज को रिलीज करना है, कौन सा एंटीबॉडीज को रिलीज करना है, कब एंटीबॉडीज को रिलीज करना है, कैसे इसे रिलीज करना है. That time, within that fractions of seconds, it starts to reproduce and we are getting the disease. This is the case when we are normal, not vaccinated. But when vaccinated, the dead microorganisms are injected in our body with an injection. That time, what our body, that is a weekend that cannot harm us, but that is a virus. Remember, so our body is releasing antibodies. उसे थोड़ा थोड़ा देर में पता चल रहा है कि इस एंटीबॉडी से जा किल हो गया. पर in the living condition when they are very powerful, when not vaccinated, that वो नहीं हो रहा क्योंकि वो तो ज्यादा पावरफुल है, वीकेंड नहीं है तो. पर during vaccination it is taking time and it is trying to kill them and it is successful, and after that it remembers everything. If the good means the virus will enter in our body, strong microorganism means the which can cause disease will enter, our body knows how to fight with it, so it will release antibody and kill it. Simple work done. This is called as the vaccination process, where we are injecting the killed microscope. When our bodies remembering that kill microscope, how to kill them using the antibodies, and when the real or the foreign particles are coming actually, that time our bodies are releasing antibodies and killing the pathogens by not creating the further diseases. Understood? Yes, it does not create more diseases. Now, disease can be prevented. Like Edward Jenner invented this smallpox vaccine in 1798. Okay, who? Edward Jenner. So we have completed the commercial purpose and the medicinal use. Now we will go to increases soil fertility. Now microbes increases soil fertility, that you all know, like cyanobacteria are there in the non leguminous plants which are not legumes. Okay, there the cyanobacteriums are there, the green algae are there, which increases the nitrogen content. You know, npk is necessary. If anyone is there, then anything can work. So it increases this nitrogen content and increase the fertility of soil, which help to the plant to synthesize the protein. So these are known as the biological nitrogen fixer, because they're fixing the nitrogen. Okay. Yes. So same way you have seen that the vegetables or the organic matters decompose or degrade in a black substance. So this is due to the action of microorganisms. They, what do they do? They degrade them. They actually eat them and degrade them and clean up the environment. Can you understand biological nitrogen fixation? What is biological nitrogen fixation? That is fixing the nitrogen in the soil by the blue-green algae and rhizobium bacteria. And next one is a decomposition, because it's cleaning the environment by degrading the organic matter. Now, how do vaccine work? Let's us read. Vaccine contains data to weaken microbial strains of particular disease. When a vaccine is introduced into a healthy body, it possesses specific cells against the pathogen. This cell becomes active when pathogen attacks to our body. Body fights with them by specific reaction. These special cells remain in the body for lifetime and protects us against the pathogens. तो जब वो एक बार क्रिएट हो गया, उसे डिस्ट्रॉय कर दिया, तो वो एंटीबॉडी सारा जीवन के लिए रह जाता है. If the pathogen will enter again, our body will treat it. Okay, it's orally means in mouth polio vaccine, right? It you are giving in mouth, so you are very small and injected. Injected means with injection. So what is vaccine? Is your first homework. Just now we have discussed it. Chalo. You can discuss within school about your friends. Okay. Now I will be taking the frequency asked out. Are antibiotics made up of bacteria? क्या लगता है? Obviously, but more precisely, you can tell microorganisms. Okay. Some antibiotics are also made up of some different microorganisms like fungi, right, mold, what we are talking about. But most antibiotics are made up of bacteria. Why should we caution in taking antibiotics? देखिए, हमारा बॉडी में आप जानते हो टू टाइप्स ऑफ बैक्टीरिया होता है, गुड बैक्टीरिया, बैड बैक्टीरिया. नाउ दिस एंटीबायोटिक्स डज नॉट नो हु टू किल. It can kill bad bacteria also, good bacteria also, antibiotic. So we should take the prescription of the doctor, what he has prescribed, and take that much doses. Okay, that much doses we have to take, because if we take excess, the good bacteria will be killed. That is not good for us. We will fall disease. Like antibodies, if they are removed, then how can we live? We will not able to live. We will not able to survive. We will not able to prevent the disease by ourselves. Our body will not get the power to resist itself. Do antibiotics kill all good bacteria? No. If used in excess, that time it can kill some good bacteria. Let's go to the quiz. Pathogenic microorganisms present in hot cells are killed by painkiller, antibiotics, antibodies. Who are killing it? Antibiotics. Pathogenic medicines, it is not telling. It is telling medicines. See the question. It will be not antibiotics. Sorry, antibodies. Chalo. Tell this one. Which are the following diseases can be cured by antibiotics? ये आपको बोलना है. डायबिटीज, टाइफॉइड, एड्स, डेंगू. एड्स तो नहीं होगा ऑप्शन. क्या मैं बोल देता हूँ, एड्स इज लाइफ थ्रेटनिंग डिजीज. caused by HIV virus because it directly सब वायरस क्या करता है, कोई रेस्पिरेटरी सिस्टम में अटैक करता है, कोई नर्वस सिस्टम में अटैक करता है, बट दिस वायरस इज सो क्लेवर that it affects our immune system. When immune system is damaged, our body can't resist any type of microorganisms because immune system is producing the antibodies. So if this is attacking the immune system, which is damaged, nothing can be happen. Okay, this is adult immunodeficiency syndrome, full form. Okay, now tell, huh? If this is there, no, you cannot register common cold. सर्दी काशी होता है फिर से आप ठीक हो जाते हो, वो आप नहीं कर पाओगे अगर आपको एड्स है, और ये लाइफ भर रहता है, लाइफ भर. चलो, टाइफाइड, वाई टाइफाइड? Dengue vaccine you don't know, diabetes vaccine you don't know, insulin vaccine you don't know. Yes, if you don't know, learn. Typhoid is caused by the vectors. Sorry, sorry, no, not typhoid. Sorry, sorry, sorry. It is caused due to water. That's why it is cannot be cured. So learn this. It was not your level. I understood. The axes are made up of weak microorganisms. Okay, chalo. Food preservation. Now microorganisms can damage our food also. We have to. That's why we have to protect or preserve our food. Right. So we have some method, that is a physical method and chemical method. Physical method like chilling and freezing, hot and cold treatment. Let me see the next page. Canning, drying, and chemical, common salt, preservative, sugar, oil, vinegar. Now let me tell you one by one each. ना आप जानते हो कि microorganisms can spoil your food. Do you know or not? Microorganisms spoil your food. Now if microorganisms can spoil your food, अगर हम लोग उसे फ्रीज करके रखें, microorganisms cannot survive in extreme cold temperature. Extreme cold temperature it is not able to survive. Also, without moisture it is not able to survive. Also, without air it will be not able to survive. हॉट एंड कोल्ड ट्रीटमेंट में बाद में आता है, इसको प्रेस्टोरेशन भी बोला जाता है. लुइस पास्टूर ने इसे इन्वेंट किया था. तो कैनिंग क्या क्या कर दे रहे हैं हम लोग, एयर पैक्ड कंटेनर में हम लोग स्टोर कर रहे हैं जहाँ पर एयर नहीं होता है, इसलिए माइक्रोऑर्गेनिज्म ग्रो नहीं कर पाता है. ड्राइंग में विदाउट मॉइस्चर, दैट्स व्हाई माइक्रोऑर्गेनिज्म कैन ग्रो देयर, कांट ग्रो देयर. चिलिंग एंड फ्रीजिंग में कोल्ड टेम्पर, दैट्स व्हाई माइक्रोऑर्गेनिज्म कांट ग्रो देयर. नाउ लेट्स लर्न द केमिकल मेथड्स, then we will see about pasteurization. Food preservation by chemical methods like salt and oil are our two main ingredients. Okay, आप देखोगे pickles, yes, इन्हें हम बोलते हैं preservatives. Oil and salt हम लोग होता है preservatives. Okay, now देखिए by common salt are मीट, फिश, आमला, रॉ, मैंगोस, टमारिट. अगर आप कोई फिश शॉप में गए हो, if you have gone in the any fish shop, at that time you have seen that all the white substance are placed over the salt. Sorry, in a fish. That is the salt. Why? Amla. Have you eat amla? अमला के ऊपर ऐसे सॉल्ट स्प्रिंकल किया जाता है. Okay, because to prevent the destroying by the this one by microorganisms. ऑयल एंड विनेगर अगर आप देखोगे तो पिकल्स, पिकल्स में मैंगो पिकल्स होता है, वेजिटेबल पिकल्स होता है, फ्रूट पिकल्स होता है, वहाँ पर ऑयल एंड विनेगर इज एडेड, दैट्स व्हाई इट टेस्टेड लाइक सॉर. It only does, it also adds taste. It does not only prevent, it also adds taste to the things. And by sugar, jams, jellies, squeeze में बहुत बहुत शुगर भरा होता है, आप देखे होंगे बहुत शुगर होता है. In a salty environment, microorganism can't grow. Same way, in a sugary environment, it can't grow. And same way, in the oily and vinegary environment, means there is no what moisture. There also it can grow. Salty environment can't grow. Sugary environment can't grow. Oil and vinegar, where there is no moisture, there it can grow. Because you know moisture's formula is H2 and H2 means water only. So water does not mix with oil and vinegar. That's why. Sometimes you have seen that sodium benzoate and sodium metabisulfate are added. Okay, like in a squash, in a cup noodles, these are added to prevent prevent the food from damage by the microorganisms. These are the artificial chemicals which are added. Chili and freezing, that was I was telling. Sorry, sorry. Heat and cold treatment. That's why I was telling. This is the pasteurization. Now see. Okay. चिलिंग इज एन इफेक्ट. अब मैं बोला कोल्ड एनवायरनमेंट में द माइक्रोऑर्गेनिज्म कैन नॉट ग्रो. That's why refrigerator is used, refrigeration is used, so that that objects can be cold in a cold temperature and so that the microorganism can't grow. Heat and cold treatment, यहाँ पर मिल्क को प्रिजर्व किया जाता है. मिल्क को 70 डिग्री में बॉयल करने के बाद सडनली चिल्ड किया जाता है रूम टेम्परेचर में. दिस बॉइलिंग जो हो रहा है वहां पर मिनी माइक्रो ऑर्गेनिज्म किल हो जा रहा है और सडनली कोल्ड हो रहा है, तो जो बचा भी होगा सडनली कोल्ड होने से वो मर जाता है. एंड ए कोल्ड एनवायरनमेंट के लिए नॉट एनी अदर माइक्रो ऑर्गेनिज्म कैन ग्रो. because if microorganism will grow, this milk will be not milk, this milk will turn into curd. That's why. Tell me which is a chemical method. Sugar, salt, a chemical method. Okay. Milk preservation, that I have told, no, दस ला इसे ही बोलते हैं पैस्चराइजेशन. देखिए द प्रोसेस ऑफ मिल्क where it is heated at 70 degrees Celsius for 15 to 30 seconds, then suddenly chilled, suddenly cooled down and stored. This prevents the growth of microorganisms. This is called as pasteurization and it was discovered by Louis Pasteur. 70 डिग्री में करते हैं तो suddenly it is coming to 10 degree and it is stored in the container. Okay. Why fresh milk फ्री बॉइल्ड? बिकॉज बॉइलिंग आप जानते हो कि एक्सेसिव हीट में माइक्रोऑर्गेनिज्म विल नॉट सर्वाइव. सो बॉइलिंग हीट कर देता है ऑब्जेक्ट को, इससे द माइक्रोऑर्गेनिज्म विल डाई एंड इट विल किल द माइक्रोब्स एंड इट विल बी नॉट contaminated. होमवर्क टू आंसर देख लेना, पर वी हैव टू एक्सप्लेन मोर. So what I will do, how and why I will write, that is the question. So I will taking the frequency based house. I have already told. I have already told I will not take anything more. Now tell me, which among the statements is not associated with food preservation? Decrease shelf life. Shelf life से हमारा क्या जाता है? फूड प्रिजर्वेशन कुछ नहीं आता है. स्टूडेंट्स फॉर फ्यूचर, ये तो फूड प्रिजर्वेशन है. प्रिवेंशन ऑफ डिके, ये तो है. एयर टाइट सीलिंग, ये भी है related. That is not related. It is not a method of food preservation, salting, boiling, drying, pickling. Pickling means adding oil and vinegar. Tell me. The answer is boiling. Boiling kills microorganisms, but it does not prevent. Okay, which are prevents growth of the microorganisms. All of this, right? It's all the chemical methods. The last topic of today is a nitrogen cycle. Is your homework three, right? So you know that there is 78% nitrogen in our atmosphere. In our atmosphere, right? 78% nitrogen only remains in our atmosphere. It does not increase or decrease like the water. See the cyclic process where the nitrogen quantity is same. First process is the nitrogen fixation where the nitrogen is converted into the nitrogen compound like NO2, NO3 like this. Biological nitrogen fixation is done by the blue-green algae cyanobacteria and the rhizobium. Okay, they convert this, and the atmospheric, yes, see, the atmospheric is done by the lightning. Okay, now nitrification is a process where ammonia is converted to nitrites. Like you know, when a thing dies, that type because we are all also eating the things, right? We are also eating the nitrogen because plants is absorbing nitrogen and it is making protein that we are eating, that we are taking, right? Now when we die, this is released, and this is again converted to a nitrogenous compound, that is Na2 and NO3, by some bacteria like nitrobacter is there, nitrosomana nitrosomonas is there. So they, what do they convert, they actually oxidize them and convert into compounds. Then assimilation, the absorption by the plants, that is known as the assimilation. Ammonification means the when we are dying, decomposition again, NH4 is coming. No, you can't understand. Means we die, that time this is released. Right. Now soil, in the soil sometimes this is only coming. That time it is nitrates and nitrate, it is converted by some bacteria. Then it is absorbed by the plants. Then again if we die, then this is only released again. That time this is called as ammonification. After this ammonification is done, nitrites and nitrates are created. These nitrates and nitrates created from the NH4 are denitrification bacteria, they are called. They do denitrification and convert it to the into gas. Okay, see, let me tell you, see how this is there. Now make it more small, you will understand. Okay, nitrogen are always like in 2. When lightning and pass, this is gone away. Now when it is mixed with oxygen, it is forming N2, sorry, NO2. And when this is mixing with hydrogen or means water, it's coming HNO3. This HNO3 is coming down, or this N2. is taken by the plants. Its N2 is taken by the plant's roots where rhizobium bacteria are present. They convert this to the compounds. Can you see? Compounds of nitrogen. Nitrification is happening, means the compounds of nitrogen is forming. Then plants are absorbing it. After it's absorbing, when the plants animals dies, the denitrification bacteria convert it into free nitrogen. So first nitrogen fixation is happening. Then what is happening? Nitrification is happening. Then assimilation is happening. Then denitrification is happening. Like that, the process is continuing. See this. You read by yourselves. Now let's go to some quiz. Sorry, doubt. How nitrogen is important for us? Protein important for us. Is NO3 is known as nitrates? Yes. N2 nitrites. Okay, that doubt cleared. Now the process by which amount remains same. What is that nitrogen cycle? Next, bacteria that converts nitrates in nitrates. Tell me. You have learned these three names: nitrobacter, nitrosococcus, nitrosomonas. Clostridium I have not learned. So this is becoming conversion. This is the actually nitrification step. Who is there in nitrification step? Tell me. Yes, correct. Is nitrobacter, nitrosococcus, and nitrosomonas are in the denitrification step. Okay, tell which enter our body. Which way the yes, yes, correct. Both, both. No, water to falling in the ground, guys. We are not drinking that water. Chalo. Let's summarize. Antibiotic is the medicine that contains microorganisms that kill or stop the growth of it. Vaccine is the weakened microorganism. Chemical method like of preservation, salt, sugar, oil. We can use sodium uh, metabisulfate and sodium benzoate. And physical methods like chilling, freezing, drying, canning, storaging, packaging, pasteurization. You have learned them. Nitrogen fixing the process which is a cyclic process where nitrogen in the air is converted into nitrogen compounds. Nitrogen cycle is a complex biogeochemical cycle in which nitrogen is formed at nitrogen. Okay. Now let me revise you for one. First is nitrogen fixation. Okay. NH4. This is coming. Now second is nitrification where NO2, NO3 these are forming. Next is ammonification. It is absorbed. Sorry, sorry. Next is assimilation, where it is absorbed by the plants. Fourth is when plants and animals died, ammonification means again it is forming NH4. Now last step is denitrification, where this NH4 is directly converted to NO2 with starting process. Like first NO2, NO3 are converted, then the gases for oxygen is released. This is the nitrogen cycle. I hope you understood, and that's all for this chapter, microorganisms. Right. Let me. So next, I am thinking to start the coal and petroleum chapter. I'm not sure. Let's see. I've completed this chapter too. Next we will see. Bye. Next day I will show you the syllabus.